Hello and welcome to Marathon webinar series. This morning we are going to talk about fish spawning aggregations in the Mar, current status and recommendation. This webinar will present the current status of the fish spawning aggregations in the Mesoamerican Reef, identifying the main threats and challenges to their conservation and offering policy recommendations to improve their protection and sustainable management. Our guest speaker is Stuart Fulton from COBE. He's the Director of Development at Comunidad and Biodiversidad. We have available interpretation service. In the Zoom controls, you can click on interpretation. Then you click on the language that you would like to hear. And in order to hear the interpreted language only, you need to click mute original audio. Some basic rules of the seminar. This space encourages learning and respect. Microphones and cameras will be disabled during the seminar. Please write your full name, country, and organization in the chat box. You can submit your questions in the chat section of Zoom. If you're participating via Facebook Live, you can submit your questions through the Facebook comments section. And the speaker will answer the questions at the end of the presentation. The opinions expressed in the webinar are the sole responsibility of the speaker and do not necessarily represent the opinion or position of Marfund. Once again, welcome, and I want to introduce our guest speaker, Stuart Fulton. He is a Director of Development at Comunidad and Biodiversidad, COBE. He has worked for over 15 years in marine conservation and sustainable fisheries in Mexico and Latin America, with a particular focus on citizen science, community marine reserves, and fish spawning aggregation. More recently, Stuart has promoted the use of technology in small-scale fisheries in Latin America to increase the impact of citizen science. Welcome once again, and thank you, Stuart, for joining us.
Thank you very much, Michelle. So yes, thank you all for being here. I am going to start presenting. The objective today is to talk about fish spawning aggregations in the Mesoamerican Reef, especially focusing on what we have worked, not only COBE, but also with our partners in Smartfish and Marfish in the late years. And here, starting with a picture of Punta Allen in Mexico, where the fishermen have worked a lot with fish aggregation. And the objective here is to show a panoramic view of all of the region. So COBE, if you don't know us, it's a association that has existed for 25 years in marine conservation and the appropriate use of resources in the fishing areas. We work in all of Mexico. And this specific project in the Mesoamerican Reef, we are focused in Quintana Roo. So fish spawning aggregation sites, as a general introduction, seeing the people that are here. I'm not going to spend too much time on this. But basically going a little bit out of the Mesoamerican Reef, these aggregations have been reported all around the world. Many of them have not been properly evaluated, and of the ones that are monitored, it's not properly evaluated. So we know that all of these sites require a lot of attention so that we can maintain healthy populations of fish. And the different types of aggregations, we know that they are massive concentrations of fish that come together with the purpose of spawning. When we talk about in a global scale, more than 300 species that have been recorded to have this specific behavior. And generally they occur in specific moments related to lunar phases and biophysical characteristics that have been identified. For example, the reef complexity and depth, and the relation with currents that may favor these spawning sites. There's different types of aggregations. We're not going to talk a lot. We're going to talk a lot about the transient ones, generally fish that migrate long distances. For example, the Nassau grouper has documented migrations of more than 200 to 300 kilometers to reach these spawning sites. And they usually take short periods of times of days to weeks in specific times of the year. And here we are talking about, well, we know that we are talking about snappers and groupers. And these there's aggregations of residents. We are talking more about parrotfish that spawn in their range, their home range, in their normal distribution reef sites. I don't have to spend a lot of time talking about the importance. Um, it's areas of large productivity focus points. Uh, it's the beginning of recruitment for fisheries. It's areas with high food productivity, and that is why they're related to synchronized large predator migrations or other animals that may feed on the fish eggs, like whale sharks. So these are fundamental sites for reef health and fishing populations. As an example, we have sites in Siancan in Mexico. We have different species that share the same spawning sites, all related to a drop in the profile of the reef. And in these sites where there is this relief and a very specific reef morphology that favors the currents and the dispersion of the larvae through time. So sometimes when we think about fishing management, we have focused a lot, and I am going to talk about no-take zones, but we have different tools of management. Here we have some of the main tools: the minimum catch size, the fish restriction, permits, quotas, no-take zones, and different fishing techniques. So we're going to talk a little bit further. Something that came up from the work that we've done in the region is that the only one that doesn't apply here are quotas. Um, for spawning aggregation, we have minimum sizes, especially the Nassau grouper. We have fishing restrictions or bans. We have limited also some fishing techniques. So we have to think that even though the no-take zones has been fundamental in the past years, in the past 25 years in the region, it is not the only management tool that is applied to manage fish spawning aggregation sites. On the other hand, the importance of large fish, and that leads us to the second part of the talk, is that a larger fish produces many more eggs in a smaller fish than a smaller fish. So that is why it's important to have minimum sizes and that the larger fish reach their spawning aggregation sites. Here in the region, this is something that we have reported in our documents that we have published lately. We have noticed that the different sizes and weights of the principal species, of the main species in the area, based on the IUCN status. And we can see two things. The larger species generally are in bigger threat, and there are more grouper species that are in vulnerable or threatened conditions. So we can see that the Nassau grouper is critically endangered. The black grouper is in Mexico. It's in Mexico, it changes name. And we have very, very little data to be able to support these IUCN status, especially with snappers, because we need more information on how the populations are behaving. We also mention the Goliath grouper, the largest fish here on the reef that was categorized as critically endangered because of stock recovery in other populations. For example, in Florida, they have reduced the category to only vulnerable, but honestly, I don't think that that is reflected on populations here in the area. There's very, very little organisms of this species that we observe in the area. So in this project of Marfish, what we have done is to basically take, after 5 years of implementation, we started in 2019 with standardizing monitoring protocols with all the different partners in the 4 countries. And from then, we developed one first report of the state of the fish spawning aggregation sites on the reef. And then we published a policy brief, a little bit of a shorter version. And these 2 documents, we just updated them because we are finishing we are reaching the 5th year of the project. So we are updating these protocols with the updated data of the region. Some of the key points, and I'm not going to go into a lot of specific data of each site, this is where all the sites are and what the stocks like in all of the areas, and if they are in recovery or if they are still declining. So we have a bit of a mix. However, we still have a lot of sites without the adequate monitoring. Because of the logistics and the costs and the complications associated with monitoring different sites at the same time in all of the region. So, as a summary, Belize is a country that has more protected areas, more known protected areas, followed by Mexico and Honduras and Guatemala because of their very small coastal area. They only have one site, but we have also advanced on the protection on this through the project of Marfish. As general data, most sites are between 20 and 30-meter depth, where we have the profile drop to the depths and reef promontories in the reef, where this complex morphology that the fish are looking for. Here we have some data on the policy brief, where we have seen that the 4 countries have their no-take times. Mexico is the time with the shortest no-take times, and we will talk later about why. In general, the 4 countries only have management tools directed to the Nassau grouper, even though we know that there are a lot of other groupers that spawn in similar sites. It's important to highlight that the measures that Belize has are very good. They have proper size limits for fishing that allows for the larger sizes, the larger organisms to reach larger sizes. And so we have a more production of larvae for the next generation of fish. For example, in Mexico, however, we have a minimum size that is very small, that is dictated from a species on the Gulf of Mexico that is not the same as a Caribbean species, but because of the management laws, they use the same sizes, and they are of course having an impact on the Caribbean. Guatemala is only one that has a management technique for snappers, This is something that has not been implemented in the rest of the region. And as I told before, the main management site is establishing the no-take zones that has been implemented in the spawning sites. As a general picture of fishing, it's not very easy to identify. These are all data from Mexico and from Quintana Roo in the Caribbean side. And it doesn't look like a very promising picture in terms of groupers. We can see in the top graph, it's the finfish, and below you can see the green line, the groupers that has a very strong tendency from 2005 to decline very, very strongly, along with all the other species that are in the list. So we don't know. These are the official Conapesca data. We know that the fish data is not reported fully. We don't really know what the situation is looking like. But there is a tendency for more than 20 years, a very worrying tendency of decline. Um, the fish aggregations are not being able to repopulate the fisheries in the region, and this is going to have very negative impacts in the future of fish that are going to come to these spawning sites. Even in Belize, where we have a lot of data, is the most complex situation, because even though good management practice were established for the Nassau grouper, however, it was a little bit too late. On the figure on the right with color, known as the Kobe plots, we can see that the fishery has from data from around 100 years ago, from not sustainable. The little white circle is year 87, and each black dot is a year. I'm not really sure how many dots there are, but about 15 to 20 years after that, when Belize actually started implementing these management measures for the Nassau grouper, you can see a tendency towards an improvement. However, the biomass in the ocean is very small. So even though there's changes towards improvements, the fact that you start with a very low biomass, the recovery process is going to be very slow. So the recovery of a very damaged and so overexploited population. And you can see on the small figure on the right, um, all of the captures that are presented are mostly immature, which means that the population is still recovering. And you can see that with data from different sites. In K Glory, we have a lot of data from fishing and scientific and articles even from the 60s that report of more than 100 tons, and always with a tendency towards a decline. And then you can see that in 2002 around, a protection site was established, but the population was already very low. So there is a general tendency to recover, but since you are starting with very, very little, it's going to be very hard. And you can also tell that with capture data from other species of groupers, where many are being captured in smaller phases or not mature phases. And so it reflects that we don't have the larger fish on the reef. And then a shift line baseline is something that we have included in our reports because the Mesoamerican reef has a lot of information about the aggregation sites. Belize was a pioneer in the Caribbean and a global level of studying aggregations since the 90s. And there's scientific reports and literature about this. And so that this way we can know how these fisheries were looking in the past. And we have this concept of shifting baseline where we don't really remember how it was before. We don't remember what it was in the past. For example, the Goliath grouper. I've been working for 20 years in the area and I've only seen three, so I can't really imagine what it looked like 100 years ago, where I imagine was a lot more populated with this species. So with the fishermen, even the fishermen can't really imagine what the reef looked like before in the times of their grandparents. Even in the scientific part, um, for example, this article of Mahawal that I show here on the right in the Caribbean, in the Mexican Caribbean, the researcher Alfonso Aguilar from the 90s has information on the site, and and basically the species was extinct around the 2000s, and now you can't really see that species. And you can revise the literature, and you can tell mm, how important this species, the spawning aggregation site, was, and they took them to Belize for commercialization. So this is from other parts of Mexico. These sites, very, very old, from William Dampier. I think he was a pirate, but he says that there was so many sharks in the fish. You couldn't even you couldn't even fish because they would eat what you captured, and other places in the Gulf of California where they talked about other um, grouper species, and they don't exist anymore, but they give you a reference of the past. Something that has been done throughout all the countries is just document all these sites and talk about the fishermen that have a lot of knowledge about this. I speak a little bit more about Mexico because it's where I know more, but it is a process that has taken place in all of the four countries, where through interviews and maps and monitoring, we try to identify these sites. So on the map, you can see all of the sites that have been potentially identified by different works, from the people in Ecosur, Kobe, and other partners, where if these aggregations are actually happening, and if they are, to implement monitoring strategies. In Mexico, we are replicating what was done in Belize before, and Belize, and I mean Honduras and Guatemala as well, to identify all of these spawning aggregation sites. So from the different reports that we have done, we have different conclusions. I think an important part is how do we define these fish aggregation sites? With groupers and snoppers are the easiest, but they are not the only species that spawn in these aggregations, and they are not the only ones that are part of fisheries. So we have identified how many species um, you need to be considered in aggregation, and all the different things that you have considered during their monitoring. Or if it's just a school of fish, the necessity to have a regional database. Um, Agra has been working on this for a more regional across borders, because we know that, of course, fish can migrate between different borders. I'm going to talk about hyperstability is a little bit more complex, where if like a fishery can sustain fishery, an aggregation can sustain fishing for a while and with no effect, and then after a while it doesn't stand it anymore and then starts reducing. So we have to be careful with that hyperstability. To highlight other ones, we know that there's areas that have decades that have been protected, and it's going to take time for the recovery to occur. These large fishes have long life cycles, so we have to be patient, and we need to estimate the appropriate amount of time for us to see the changes. And the last one I want to highlight is the climate change generates a lot of uncertainty. Talking from experience, for example, here in Mexico, if we talk to the fishermen, to the older generations, they have told us that the aggregations of groupers, they started forming since December, and we have been looking for them in December for 10 years from with diving or with hydrophones, and we just can't find them. So apparently, so there's been a change, possibly because of the changes in water temperature. Um, the spawning time has moved a little bit to more in February and March instead of December and January, as we could see, as they reported in the past. So, yeah, the no fishing times have been established based on these historical data. Here, I'm not going to read everything, but the key recommendations, I'm going to speak about each country in a little bit. Um, we know that fish cross borders, and the management should be the same in all four countries. The larvae distribution move and the benefits from other countries benefit from what may happen in another country. So we need coordination in a regional level, and even decision making between all four countries. Uh, uh, Efficient data management. Uh, every time it's very expensive to go out to monitor, and sadly, it happens that the data is lost or that they damage. So just make sure that this data collection efforts are continued throughout time. And this is what we are trying to achieve with the different collaborators. I think the other ones to highlight, just to keep all the monitoring efforts in the long term, try to have adaptive management wherever you can. But we know that this changes, or that no take rules or quotas is not easy to change them. And always to try to invite the fishing sector to participate, because they know what they're talking about. They know, and the government doesn't really have money to be participating and taking care of that. So we need to push for that community participation. And just to start closing, the recommendations for countries in Mexico. Mexico doesn't recognize fish spawning aggregation sites as important sites. It is not in the fishing legislation, and that has large implications on the whole Caribbean, because um, there are different reproductive behaviors um, in the Gulf of Mexico and in the Caribbean. So we need to have more tailored management to the Caribbean and from the Gulf of Mexico. It doesn't mean that we change things in the Caribbean that might affect the people from the Gulf of Mexico. So we need local laws in the region. And the same for the minimum catch size, because we are using a species of grouper that doesn't exist in the Caribbean as reference, and that doesn't make sense. It is important to evaluate and understand the snapper fisheries and what is being done uh, with these species, and we need to have data to be able to base decisions on this species in particular. And focused on the marine protected areas, we need to revise and update the management plan, especially for the aggregations that are so very important. And uh, Belize already has a lot of measures established. And so, for example, uh, focusing on the Nassau grouper, but there are a lot of aggregation sites where there are fishing permits in the site, which complicates the management of the site. So uh, you need to extend the regulations for other species. So apply all these measures that have been established for the Nassau grouper that are mostly almost perfect for only this species. So make that also stand for other species of groupers and snappers. Um, think about how to align the management of fisheries in Belize and take into account the different species that are being collected um, at a regional level, improve the vigilance and replenishment of the areas, and empower the local fishermen and improve technology for the monitor, something that is in a pilot state in the area. In Guatemala and Honduras, try to implement more management strategies, especially in Guatemala. It's all four all four countries need to increase their monitoring and their vigilance to be more protective over the spawning sites and to search and to try to have more data on the spawning sites in Honduras. Before we had mentioned all of that applies, but also create more no take zones in different sites and and try to have better management sites throughout time uh, because some sites are not validated in of the fish spawning sites. And with this, I'm done. I don't know if there are any questions or doubts or any other comments. Thank you very much, Stuart, for the presentation. We open the room for questions or comments. You are welcome. You can activate your microphone or you can write it on the chat so you can ask your questions for Stuart. Okay. I don't see any questions so far, Stuart. I don't know if you have any more comments or anything to close with. I think I need to highlight the importance of projects like Markfish for being a project in the four countries and a lot of collaboration with different groups. I think we need to search to look, and we need to look to work at that level. You can't just focus each one in their area. We need to have a regional collaboration. Okay. Yes, thank you, Stuart. Stuart has based his presentation in two documents. The document of aggregation policy and the current state report that you can find on Marfund and marfish.com. So you are welcome to visit these documents and learn a little bit more about this subject. And maybe if you also want to, if you missed the first part of the presentation, you can also access the presentation on the webpage on those links. Here we have a question from Patricia Kramer. She says, you mentioned that there is a high potential on the effect of climate change on fish. This change in time, does that change in the timing or of where and when to monitor? Yes and no, we don't know. More in the time. It seems like because we are not 100% sure, apparently it has moved. The initiation of the spawning has changed from December. It has moved to January or February. The where so far, not really. The sites that we have documented before, the site hasn't changed. The fish are still there every year, but but yes, it does appear that to have shifted in the timeline. Thank you very much. We invite you all to see this document uh, in the status report of the fish aggregations and the policy brief. And we also ask you to fill the document that has been posted on the chat so that we can improve our webinars. And if there are any more questions, I thank you all again, and then we can begin a closing. Oh, there's another question from Patricia. Are you currently tagging and monitoring, and any insights from the work of migrating patterns? No, we don't. We only have passive acoustic monitoring. We don't tag traditionally for the fish. That would be very important, very interesting to be able to understand a little bit more their migration uh, in in the level of the mar. But a very interesting site with the acoustic monitoring that we've done, we have documented that the fish that we have in the region have have different frequencies uh, to other sites in the Caribbean. The very little information, genetic information that we have is that the fish in the Mesoamerican reef has what it looks like a sub population with slightly different genetic um, information from the Caribbean, and we do need more information on how they're migrating and also how much retention of the larvae are we getting, or where they are being distributed, or if the Mesoamerican reef depends on other sites in the Caribbean for the availability of larvae. But that would be important to address in the future. Well, again, thank you, Stuart. We give closure to our presentation, and thank you all for participating and their interest in this subject. Thank you all so much.